that's really grainy still, which is not great. Hi, if you're just joining me, apologies, I am just trying to load up the session. If you have specific questions, please do feel free to ask away. What am I going to do? I was going to carry on with the paper 2023 paper 1 that I had started, and we are going to do that. Okay, so I'm hoping that this will, there we go, go from there. So if you are joining me, welcome. Sorry, I'm just trying to get this sorted. There we go. Okay. So this was the latest question, and it was looking at you understanding about the communication system. So let's have a look at the question and see what it says. So we have got, which statement about communication systems is correct? Cell signaling acts only over short distances. And remember, whenever you are doing multiple choice questions, the best thing you can do with these multiple choice questions is know how to actually eliminate the wrong answer. So if you can eliminate the wrong answer, you are going to help yourself to figure out what the right answers are. Absolutely, there is still time to go from a B to an A star if that is what you're looking to do. It's really important that when you do the work that you are trying your hardest to make sure that you are limiting the distractions that you may have in your life. That's one of the hardest things. I'm going to see if this makes it a bit better. I'm not sure if that's going to help anything on YouTube, but we'll see. Okay. Not really, but never mind. Right, here we go. So, which of the statements about communication systems is correct? And we want to look at the information that they've given us and eliminate those wrong answers, leaving us with the only right answer. So, cell signaling acts only over short distances. Well, no, it's not going to only act over short distances, because we know that a hormone, for example, that is released from the pituitary gland, ADH, is going to make its way all the way down to the kidney collecting ducts, where it's going to have the action that it's going to do. So therefore, I know that that is not a true statement. So we can eliminate A, because I know that it's not over short distances. So now we need to think about, well, what else could it be? Is it on B? I have no idea what's going on with that. There we go. Hi guys. I know, Callum. Do you know what? I've been so ill. My delightful youngest daughter decided to give me a cold, and so therefore I was really ill last week. So I completely shut down. I did obviously my clients, and then after that I just had to have some time to myself because I just felt so rough. I'm still not 100%, but I'm better than I was. So I thought I'd jump back on here. Okay, so, oh, thank you. It is, it's definitely going around. And it seems to be hitting, for example, me harder than it was hitting my youngest. She was like, oh, I don't feel very well. Whereas I was like, oh, I feel like I'm dying. My pain is just so bad that I was taking painkillers. And then I was like, oh, so I'm taking painkillers, and the painkillers are binding to the receptors and they're stopping the impulses. So obviously I went down the whole biological route side of things. But anyway, right, let's get back to this question. So communication systems are required to coordinate responses and maintain a near constant internal environment. Well, that kind of seems right. Because, yeah, that's the whole point in communication systems. Our cells are going to signal so that we are maintaining homeostasis. So I think that one's right. But let's just double check, because if that one's right, that means I must be able to eliminate the other wrong answers. Effectors that respond to hormones do not respond to nervous stimulation. Effectors that respond to hormones, well, that's a load of rubbish, because if I think about my sinoatrial node, that is going to respond to adrenaline and also respond to my para and sympathetic nervous system. So I know that that's not true. And so therefore it's important that you are eliminating, as I've said, because then you are ensuring that you've got the only right answer left. Okay, plants do not require communication systems because, unlike animals, they do not respond to the environment. What? What a load of rubbish. Plants do not require communication systems. Well, if they don't require communication systems, why do they produce hormones? For example, why do they produce ABA in the response to water stress? So we know that that's not true, which would leave us with the only right answer, which is what we thought it was. So it is important that when you're doing these, that you're practicing, that you are looking at what is the question actually asking and how can I ensure that I've got the right answer. Okay, let's go to the next one then. So for those of you who are just joining me, this is paper 23, paper 1 from OCR A that I am going through. So I am going through how exactly I would eliminate the wrong answers and also leaving you with the only right answer. Okay, so the Pacinian corpuscle is a type of sensory receptor. I love the Pacinian corpuscle. I hate saying it, but I do love the Pacinian corpuscle for what it does. So which of the options describes the action of a Pacinian corpuscle? So a strong stimulus at a Pacinian corpuscle produces a generator potential that exceeds a threshold value. So if we're thinking about this and we're thinking about, well, what is a threshold value? So if I were looking at a graph and I was looking at the potential difference of my membrane, I know that I should have my resting potential. I know that I depolarize my membrane, and if I go past the threshold value, that will therefore cause an action potential. And an action potential is depolarization, repolarization, hyperpolarization, and then that brings me back up to my resting potential. So a strong stimulus at a Pacinian corpuscle produces a generator potential that exceeds the threshold value. I think that one is true, but if that one is true, then I need to eliminate the wrong answers. So to be able to eliminate the wrong answers, I need to check a strong stimulus at a Pacinian corpuscle produces a larger action potential. No, it's not a larger action potential because the action potential is an all or nothing principle. And so therefore, an action potential will occur, depolarization, repolarization, hyperpolarization, and then back to resting potential. So I know it's not a larger one, so I can definitely eliminate B. Okay, C. Pressure on the Pacinian corpuscle causes the neuron membrane to become less permeable. Well, absolutely not. It becomes more permeable to sodium ions. Because it becomes more permeable to sodium ions, the sodium ions are going to cause that depolarization. And so that is wrong. The Pacinian corpuscle is a transducer that converts pressure into chemical energy. No, it doesn't convert it into chemical energy. It converts it into electrical energy, and therefore that would make that wrong. So A was my right answer. So it is important that you're looking at how to eliminate the wrong answers and not just automatically assuming that you've got the right answer, because sometimes you've guessed it, sometimes you know it, and that's great. But if you cannot actually explain, oh, I just put that one in the wrong place. If you can't explain the reason as to why the wrong answer is the wrong answers, you don't know the topic area. So it is really important as you're going through this to make sure that you are able to see and describe all of the different parts. Okay, so let's look at the next one. Oh, no, there we go. Okay, so guys, thank you for the likes. Thank you so much. If you want to, I am currently live on my other channel, obviously. So please do pop over if you want to see the questions. I am trying to respond on both. Nobody's chatting on YouTube. I'm not sure if there's something wrong with, there's a delay there or not. Okay, this question then, the next two questions that it's telling us are going to be related to this graph. And so one of the things I always say to my students is take your time going through the questions. The reason I say take your time going through the questions is you can sometimes read the information, skim read it, move on, and not have digested what it's telling you. Also, for those of you who've never come across how I do what I do, I look at these questions and I always think about, well, how could they use this in a different way? It's never just about looking at these past papers and memorizing them, because that's not what they're going to do. They're going to give you a whole new set of questions, but the science, the biology hasn't changed. And so therefore, where else could this go? What else could they want to talk about with these questions? So do take more time in your revision to look at what they've given you. Is there a live every Monday? No, not normally. Every Wednesday is when I do normally go live. Monday's a hit or miss. And because I didn't go live last Wednesday because I wasn't very well, I decided I would jump on here tonight before I send my session to my group tuition lot. So my video is downloading from the session I've just done, and so therefore that's why I thought I'd pop on here. Right, let's look at this question. So beetroot cells contain a red pigment. So we're being told we've got these cells, and these cells contain a red pigment inside a vacuole. So this is where I would be saying, okay, I know a vacuole is a membrane that is made up of my phospholipid bilayer. And so for those of you who've just come across me as well, what I'm doing right now is I am activating my brain to try to get to a point where I've thought through all the links that could be coming up so that I can then talk about how it relates. So phospholipid bilayer. And sometimes they do tell you bits later on, and that's great, but I like to take my time going through because then I know that I've thought about it in the right way. So it then goes on and says some students are investigating the effect of ethanol. So we've got ethanol on the permeability of the plant cell membranes. Now, again, they haven't actually said the phospholipid bilayer, but they're expecting you to have made that connection already. The other thing they're expecting you to remember is that you have got what's known as the fluid mosaic model. And so do you understand that the fluid mosaic model is about those phospholipids and those proteins and the fact that they can move around within the membrane? So what have they told us? This method that they used was they soaked individual pieces of beetroot for 30 minutes in different concentrations of ethanol. So again, what I'd be saying is as I'm looking through this, I'm going, okay, what could they do? Well, they could turn this into a method question where they're assessing module 1. What's the independent variable? What's the dependent variable? What are your control variables? So sometimes they give you lots of information in stem of the question, sometimes not. This is a multiple choice question. They're not going to give me lots. However, I would say thinking about it this way is going to help you. So they've said soak identical pieces. Well, how big were those identical pieces? Would be what's going through my head if I were asked to evaluate this type of question. Identical pieces of beetroot at different concentrations of ethanol. So I know that my independent variable was different concentration of ethanol. I would hope as a control variable that these identical pieces of beetroot have the same size. Now the fact that they have said identical pieces, that is implying that this size has been kept the same, and that therefore is implying that it is a control variable. Is A-level biology hard? Yes, it is, because there's lots of content and you have to keep on top of it. However, it's actually really enjoyable if you are interested in it. Sometimes it becomes more challenging when you are being taught things that are boring, when you're being taught things that are really challenging. So it's a subject to choose if you enjoy it, because you have to work at it. Would be my biggest advice. Okay, after 30 minutes, measure the redness of the ethanol solution using a colorimeter. So therefore, they're assessing my understanding of colorimetry, and they've told me that this red pigment has potentially leaked out of the vacuole in the beetroot cells, because they're looking at how much redness is in the ethanol solution. So again, what is the detail that they've told you in the question? How can you relate that? Because you should know that you have got your, let's say they take a beetroot and they have it as 1 by 1 by let's say 3 centimeters. So you've got a piece of beetroot. They told me it's got a red pigment. Well, it's more purple, but anyway. And so therefore I've got my red pigment. They've then told me that they've placed it into an ethanol solution. And so if they've placed it into an ethanol solution, that ethanol solution, let's just colour this in. What they're testing when they are testing it after the 30 minutes is they're testing the solution. So therefore that red pigment must be leaking out of the beetroot. And what you're assessing is how much of it is leaked out based on the different concentrations. So you've left it for 30 minutes. So they have probably done a preliminary investigation to know that that's the right time. And then what they've done is said, right, here's my graph. And in the graph, I've told you that plus or minus 2 standard deviation are being shown at each data point. So they're giving me so much information, and I know I haven't shown you what the question is yet, but I would therefore say, if they gave this not as a multiple choice and they gave this instead as a, could you evaluate the validity in the results that have been obtained, do you know what to talk about? Do you know why it's important that they've said that you've got the mean plus or minus 2 standard deviation points, and where these error bars are actually giving us, if I were to draw my line of best fit going through the data points, what does that say? Well, I can see as I increase the concentration of ethanol, I am therefore seeing an increase in the absorbance of the solution using the colorimetry. So I'm describing the data and I'm trying to draw a conclusion based on what I can see. So therefore my assumption would be that as I increase the ethanol concentration, I'm therefore going to assume that it's going to disrupt the phospholipid bilayer, and by disrupting the phospholipid bilayer, that pigment is now able to leak out. Because remember, the pigment was contained inside the vacuoles, therefore the pigment was too big to get out across the phospholipid bilayer because it was contained within the vacuoles. Only if I have disrupted the phospholipid bilayer of those vacuoles has that pigment now managed to get out. So let's actually see what the questions were. So these were related to 2 questions. Let's start with question 14. So if we go to question 14, then we're being told, one student made the following observations. So ethanol disrupts the cell membrane, leading to increased permeability. So do we agree that it is going to disrupt the cell membrane? Well, we've already said that we believe that it does. graph plateaus as all of the butylene leaks out. Right, yes, that is exactly one of the things that I didn't mention, we didn't talk about before. If we go back up here, why are we getting this sort of plateauing? Well, it's plateauing because all of the pigment has been able to leak out and it's now got to an equilibrium. And because you know that molecules move by diffusion, that if there's something not stopping it, it will be able to diffuse until it reaches an equilibrium. And we can see that absolutely in our graph because of the way that it's plateaued off. So let's just have a look. The permeability of the cell membranes is proportional to concentrations of ethanol between 0 and 50%. So let's go back and just see what that's saying. So it's between saying between 0 and 50. So this next marking point, which I'll come back to, between 0 and 50, it's saying it's proportional. So if I go back to here, the permeability of the cell membranes is proportional to concentrations of ethanol between 0 and 50% ethanol. So between 0 and 50, it's proportional. Well, we can kind of see that that's showing a bit of a straight line there. So therefore, I would say it is proportional. And so I would say that that is also a true statement. So the last thing we need to look at then is number 3. The cell membranes are completely disrupted in 60% ethanol. So if we go back to our graph here, are they completely destroyed at 60? So if I go to 60, we can see that it hasn't yet plateaued. And so because it hasn't yet plateaued, we haven't completely destroyed the membrane. So that therefore would mean that that one is incorrect. So if we come back down here and we say that one's not true, so 3 is not true, 3 is not true. We've already got rid of D because we know 2 is true. So therefore, we've left ourselves with the only right answer, which is B. And so by taking the time, we'd already primed our brain to look at the information and to decipher, which, although it feels like you spend a lot of time on the question, but if you do spend a little bit longer, you will actually be quicker later on trying to answer the question. Hi, what we're doing, we are looking at paper 1 of 2023. So I'm currently going through the multiple choice. I'm just about to finish the last question on the multiple choice point. And this is about statistical tests. So you should know the 4 statistical tests. Chi-squared, Spearman's rank correlation coefficient. You should know that there's 2 t-tests. Now they haven't specified which t-test. They've just said a t-test. So there are 2. There is the paired and there is the unpaired. The standard deviation is looking at the variability in our data. So what we need to do then is look at, well, what have they told us in the question? Do make sure that you take the time, that you read the content. How do I memorize topic 7? I'm struggling so bad to remember everything. So you are, topic 7 means that you are AQA when you say topic 7, because there's only 6 modules in OCR. I can't remember off the top of my head what module 7 is. Is that the mutations, biotechnology, not biotechnology, DNA sequencing, etc., all that stuff? This is where I need to open up the spec to look and remind myself. So electrophoresis, oh, that one. Okay, so you need to go to the population ecosystem stuff. That is in my 6.1.3, I want to say. But that is a case of you have to just learn it. But not learn all of it off by heart. You need to learn the parts that you can apply to whichever example they decide to give you in the exam. But also do be very aware your Simpson's index equation is different to the one that's OCR, so do make sure you're using your specification on that one. Okay, back to here then. The students were asked to decide whether the permeability of the membrane is significantly greater at 50% ethanol than 40% ethanol. So we're looking at two different concentrations, 50% and 40%. So because we're comparing the means of two sets of data, that automatically tells us that we're looking at a t-test. A chi-squared test is where you would have observed versus expected data. So you would have to be able to work out your expected data. We know it's not that. You have Spearman's rank correlation coefficient. What we'd be doing with that one would be if we were actually looking at, does the concentration of ethanol affect the absorbance of the solution? So if we're looking for a correlation between one and the other, then we would therefore be doing a Spearman's rank correlation coefficient. They're not doing that. They've been very clever in what they've asked. They've asked you for 50% ethanol and 40% ethanol. So therefore it has to be a t-test, and it would be an unpaired t-test, not a paired t-test if they were to take it a step further. So they could give you a graph like that again and then ask you to actually do the calculation for the t-test, or they could ask you to do the Spearman's rank correlation. It really just depends. Don't get hung up, and this is the bit that worries me when students memorize. When you see a graph like this, you're like, oh yeah, I recognize that graph, but that graph could have come up in 3 different exams in 3 different years and used in different ways. They just use the same graph, and that's one of the things that you have to be aware of. Memorizing and being like, oh yeah, I recognize that, doesn't necessarily mean that you're seeing the exact same question that you had. So when do you use the paired and the unpaired t-test? So that would be a paired t-test as if you were looking at something with the same individual. So if you're looking at, for example, me, and I am measuring my heart rate whilst I'm sitting down here, or you're measuring my breathing rate whilst I'm at rest, then you say to me, okay, go for a gentle walk around the block. So I go for a gentle walk around the block, takes me like 5 minutes, maybe not so much. I come and I sit down and I take my heart rate again, I take my breathing rate again. I'm the same individual, okay? So because I'm the same individual who's done it, and I'm comparing the difference between the start, the rest, and that of the activity, that would be a paired t-test, because it's about the difference that has occurred within my heart rate or the difference within my breathing rate. So if it's the same individual, it's a paired t-test. If it is an unpaired t-test, there is no connection. So in this case, it was asking about the 30 or the 40 and the 50 degrees C. No, it wasn't degrees C because we're not talking about temperature. A concentration of ethanol, 40 and 50% concentration of ethanol, they were different. So they soaked equal pieces in different concentrations. So they weren't the same piece in the same in different ones. So I hope that helps answer that question. Can you revise just by doing many past papers and mark schemes? Yes and no. So past paper questions are amazing because they will help you to be able to see where the exam boards are going to go. Mark schemes are good because they give you an idea as to what was accepted the year that the student sat those exams. But one thing I would say to you is note that the mark schemes will change based on the year and the examiners who are sitting marking your papers. So just because they've accepted it 1 year doesn't necessarily mean it will be accepted again. So the allows, etc., they will be determined by who's sitting the exams or who's marking the exams that year. So be aware of that, and therefore I would say to you that it's important you take the time to learn the content, learn how to use the papers to your advantage rather than just memorizing mark schemes. Okay, let's move on. Question 16. So we're still looking at paper 1. We're still looking at 2023's. Now, just to be aware, whenever you're going through these papers, it is good to look through them, and I have gone through them myself, and I've gone through all the papers from 2017, even below, down to 2014, 15, etc. But what I would say to you is, if they asked a question one year, for example, 2023, they've asked about synapses, does that mean they're not going to ask about synapses again? Absolutely no, not at all. What it means is they're not going to ask the same questions about the synapses, but they could ask another question about synapses, which is still relevant and still assessing your understanding. So be aware that if they asked something last year, does that mean they're not going to ask it again this year? No. It means that they won't assess it in exactly the same way, but they may. For example, if they've assessed something in paper 1 this year, 2023, they may assess it in a paper 3 in another year. Who knows? But it's something to definitely go through. Yes, thank you. That's absolutely right. We are looking at the synaptic cleft. So it's really important when you are doing this that you are actually writing things like the synaptic cleft, and what you'll find in the mark scheme is the cleft will be underlined. So if you wrote synapse, you would not get the marking point. I talk too much and I end up drying out my throat. Right, okay, let's carry on with this question then. Identify whether structure A or structure C is the first neuron in the pathway. How would we know? Well, whenever we look at a synapse, we always need to think about the fact that we have our presynaptic, so there's my presynaptic neuron, and this is my postsynaptic neuron. And so therefore, if I'm looking at my presynaptic and my postsynaptic neuron, what would I expect to find in my presynaptic neuron? Tony, how are you doing? How's uni? I hear from Paul you're doing quite well. I hope you are. Feels weird not having you come in my sessions all the time. Saying that, Chidima's not been here tonight actually. A is the first neuron because you see exocytosis taking place. Nice. Very nice. So we would expect there to be exocytosis where we have got the vesicles containing the neurotransmitter are going to fuse with our membrane. So we would expect there to be the release of the neurotransmitter from the presynaptic neuron. Wouldn't there be neurotransmitters released? Yes, they are, but in a case of, in this example, you can't see them. So it's about being able to explain what it is and why you think that. So we can quite clearly see that we've got our exocytosis evidence that's gone on in 3 different locations, actually 4 if we include this one. And also we've got all our vesicles in the presynaptic neuron. So another thing to note is the evidence doesn't have to be just one way. So one of you might have said, as you did, the exocytosis has taken place, whereas someone else might say, well, the presynaptic neuron would have the vesicles and I can see the vesicles. So that would therefore mean that C must be the postsynaptic neuron. So what would we expect to find on the postsynaptic neuron? So we would expect to find on the postsynaptic neuron are receptors for the neurotransmitter. So again, what am I doing right now? Yes, I'm answering the question, but I'm also then just double checking, what else do I know? What else could they ask me if I carry on? And so it's about priming yourself to help you moving forward. Okay, let's look at the next one. Okay, so the next one is complete the sentences about the action potential. The action potential involves rapid opening of voltage-gated sodium ions. Yes, this is OCR A specification, and this is 2023's paper. paper 1. And whenever you are using other exam papers, it's okay to use other papers like AQA or Edexcel and B, but only do that if you know your content and you know what is needed or what's not needed. Because if you don't, what could happen is you could start learning something that's irrelevant to your examination, and then you've wasted time. So if you have done all of the past papers, go back and do them again, or use other ones that potentially are out there. Yes, good. So we have the fact that it's positive feedback and that this one is depolarization. So the action potential involves rapid opening of voltage-gated sodium ions. This is an example of positive feedback. So my question would be, that's great that you've been able to tell me that it was positive feedback, but can you explain why it's positive feedback? What is positive feedback? Because this is where students will have seen this question before, and students will then go, oh yeah, I know this. I feel really positive about this. I'm so good. I get this. It's positive feedback. I memorized that one. And then they switched the question around and said, explain how the rapid opening of the voltage-gated sodium ion channels is an example of positive feedback. And could you answer that question? Because the change is being enhanced. What change? What change is being enhanced? Do I have any advice on the plants and animal responses? So the plant response, I have a study guide actually on my website which covers plant response, and I took quite a lot of time going on to that one. So I would say go and have a look at that and see if that helps you. Obviously, it is a paid resource, but that would be my advice. Animal responses, the muscles ones there, I haven't done the brain yet. I'm about to launch the plant cloning and animal cloning booklet. Probably tomorrow I'll finished it, just double checking it before I upload it. And then the second part of the animal response would be coming. But if you haven't checked out my videos on the playlist, then do check out those videos. They are there to help you, and hopefully they'll make it easier for you to understand. More sodium ions are entering. Yes, it makes it so that potassium ions counteract this change by making its voltage-gated potassium ion channels being opened. Be careful. Be careful with regards to, we're talking about positive feedback. So you're absolutely right. Change is being enhanced. When we talk about positive feedback, if we have a stimulus, and in this case, the stimulus is the depolarization of the membrane. So when we have the membrane depolarized, the minute we open those voltage-gated sodium ion channels, sodium ion floods in. So what we've done is and you've used the term enhanced, we have increased the effect of the stimulus. So you've said enhanced it, I will accept that, but what we're doing is we're increasing the stimulus rather than reversing the change. So because we're increasing the magnitude of the stimulus, we're making it more depolarized. That sodium flooding in causes the depolarization of the membrane to become greater. That is why it is a form of positive feedback. Now, you mentioned that it's the potassium ions counteracting this by the voltage-gated potassium ion channels opening. That wouldn't be positive feedback, because that would be the reversal, wouldn't it? So that isn't an example. The sodium ions flooding in is the example of the positive feedback. So whenever you are going through this, always try and see if you can add a bit more to your notes to check if you understand it. And that's easy for me to say because obviously I've been teaching this for many years and I know where it could go. You are very welcome. This is one of the things of why I do these sessions when I do them, is because I want to answer and help you with the questions that you have. And obviously my paid stuff comes first, but I am trying my best to give you what I can, because I know not everyone can afford it. And it's also why I've put out there my study guides as well, because for me, I am a very visual learner, and so therefore I find sometimes I need to break it down and really understand it. Otherwise, I won't be able to explain in the exam what it means. Jake, not tonight, but I'll put it on my list of things that I will do, because it's something I think, although it's not, the calorimetry is not coming up as much for OCR. I do feel like ecosystems is going to come up a lot with regards to energy transfers this year. So if you are watching me and have been for a while, these are random little snippets of where it comes out of my brain, and I think, ooh, I feel like that's coming up. Now, this is a guess. This is obviously I've done my research and I've done lots of analysis on these papers, but it's still just my guess. I am guessing what I think they're going to do. I might be right, and if I'm right, great, that's amazing, but don't put all your eggs in one basket and believe that I'm going to give you what's on the paper, because I don't know. I won't know, and I will make some mistakes if I sit this paper like you, because I won't know what those examiners are looking for. So be aware that your teachers are trying their best, but nobody knows apart from the person who's written them. And that would be my one thing I would say. So lots of practice papers are amazing, but don't believe that a predicted paper is definitely going to be what's the right thing out there. Okay, so you're saying this one's looking at cytoskeleton. Cell movement is one, right? Acts as a path for vesicles to move, and mechanical strength. So this is an interesting one when you are looking at the questions, that you are making sure that you know what it is it's actually asking you. So mutations in genes code for proteins in the cytoskeleton have been associated with several diseases of the nervous system, including neurodegenerative disorders. Give 3 functions. Absolutely. Shape of the cell, so cell structure, the ability for the cell to move, for the movement of organelles within the cell, for the microtubules, allowing for cell division. So there's lots of different ways that you can go on the cytoskeleton, and it was one of the things that I noticed more and more was coming background, that they really want you to understand that everything within the cell, if you think about, if someone mentioned about animal response, if you think about the muscles are made up of myofibrils. Myofibrils are organelles with these proteins inside. These proteins which are allowing for the contraction of those cells because of the sliding filament theory. Well, the cytoskeleton is made up of these proteins which allow for the cell to move. So it's about the differentiation of the cells as they go. Yeah, it's an interesting one. A lot of the questions will reappear, and it's just about whether they're asking, in this case, they've linked it to neurodegenerative disorders, or they might link it to ciliated epithelial cells, or they might link it to plant cells. It just depends. But when they're talking about the cytoskeleton, they're looking for you to know the same parts, the same answers, but just relate it to the bit that's in front of you. Okay, so give me a second. Where have I gone? So this next one then, let's look at this one. Okay, so suggest how a mutation in cytoskeleton genes could cause a disease of the nervous system. So what is a mutation? So this would be what I'd say. What is an actual mutation? Do you know what a mutation is? Can you describe what a mutation is? So if you can describe what a mutation is, can you then use that to then explain how that could cause a disease of the nervous system? Now, interesting, mutations, isn't mutations module 6? And yet I'm doing a paper 1. So why is this question paper 1 when we're talking about mutations, and mutations are in paper 2 or paper 3? So why is it that this is here in paper 1 when we're talking about mutations? Well, you should know as part of module 2, nucleic acids and nucleotides, that during DNA replication, spontaneous mutations occur. It's not asking you about, is it a mutation that is a substitution? Is it a mutation that is a deletion? It's not asking, is it a mutation that is an addition? It's asking you how a mutation could cause a disease of the nervous system. So it's asking you, do you actually know how you can eliminate the wrong answers from here by using what you know on module 2? So if you know module 2, if you know about the changes that go on, you should be able to answer this question, which is, if I'm going to change, if I'm going to have a mutation, that mutation is therefore going to change my amino acid. It's going to change my amino acid structure. That's going to change my amino acid structure, which is going to therefore change my protein's primary structure. If it's going to change my primary structure, that could therefore change my tertiary structure. And if it's going to affect my tertiary structure, what is that then going to do? That is going to then affect the way in which the cell can function. Right? That's what it's basically saying. It's saying, can you understand the difference between one thing and another? One second. Oh. My computer is playing up on me. Oh, it's telling me my stuff is ready now. I just need to move some things over so I can send an email to my tutees in my group tuition. So bear with me 1 second, guys, whilst I do that. And then I will carry on. With this question. Oh, excuse me. Right, let's go back to the question. Is it because it would change the function of the cytoskeleton as its protein structure would change? So you're absolutely right when you're talking about the change in the protein structure, and then you're talking about the fact that that would then affect the protein's ability to be able to function. So in this case, what are we looking for? We're looking for, sorry guys, give me 1 second. This is being a little bit temperamental on me tonight for some reason. Oh, I think it's getting close to your bedtime. You're still not well, Christine. We're going to work slow. I have to update my laptop actually because my graphics card is not good enough for my diagrams. It keeps crashing on me because I've got too many diagrams for my study guide, so I need to update my my laptop, not paper, because I'm not using paper anymore. So yeah, I have to sort that one out, which is a bit of a frustration on that front. So if anyone can suggest, my partner keeps saying to me that I should be using the a Mac, and I'm like, I don't know. I'm so used to using a laptop, and so therefore, do I really want to use a Mac? I'm not used to a Mac. Okay, so changing the structure of that protein, because that's what we're talking about. What is that going to mean? Well, what do we need with our nervous system? Our nervous system is about our neurons being able to have that electrical impulse go down, so the action potential make its way down the axon. What else is going to happen? Well, we need for our vesicles, which have our neurotransmitters, to be able to move and fuse for exocytosis to occur. So therefore, what do we require to happen? We need for them to allow for the movement. And remember, we said the cytoskeleton was about moving the organelles around. It's about allowing the cell to move. So therefore, if we have a mutation, we're going to change the structure of these proteins. We're therefore going to affect their ability to move those vesicles with a neurotransmitter. We're not going to be able to have the synaptic transmission that we need, therefore affecting that nerve impulse and how it's going to move around. So it's really important. By that I'm referring to the primary, secondary and tertiary structure. Yeah, absolutely. So sorry, I was just doing a little thing there. Is the cytoskeleton made out of protein? Yes, it is. It's made out of proteins, which are things like tubulin, and it's to do with genes. Yeah, you're absolutely right. So the fact that you've got a gene here means that that gene is going to be expressed into the transcription translation occurring, and therefore you are having that primary structure affecting the secondary structure. And yes, it is a 2 marker. So it is a 2 marker. Let me just make myself a little bit smaller. There you go. And okay, right, let's move on to the next question. Oh, you gotta love the next one. I know, I need to download this paper again because this has given me different things. Hold on. Seems to have missed 17 and 18. One second, guys. Let me just download it again from OCR because my copy that I have is missing parts, which is not good. Where am I going? There we go. 2023, paper 1. Yeah, that was actually question 18. Now that now makes way more sense. So I've totally missed out a few questions. Hold on. Let me just... So we finished 18. Let's do 19 anyway. And then I'll come back to questions 16 and 17. There we go. Right. There we go. Question 19. Right. What have we got? We've got homeostasis and we've got excretion. Now, I do have a feeling, again, another thing where I believe homeostasis may come in quite heavy this year because it's been a while since they've done any assessment on that. Now, how they ask it, who knows? But the way in which they ask it, I feel like potentially the hormonal system will come in with homeostasis. Not excretion, but the hormonal system. But again, I may be wrong. Okay, let's look at this one then, and then I'm probably going to finish for the evening. But I will be back on Wednesday, so thank you if you are joining me. So you're saying let's go true, false, true, true. Now I haven't looked to see if you are right. I haven't read it yet, but that's what you've said, so we're going to go with it. Yep. Thanks everybody. Good night. So one of the things I would say whenever you're doing this is your initial instinct that you have to go true, false, false. What you need to do is you need to make sure that you are double checking that you have the gone through and analyze whether it is right or wrong. What is it that makes it right? What is it that makes it wrong? How are you able to get rid of the different parts? So do make sure when you're doing it that you are looking at those bits of information. So the liver, kidney, lungs and skin are all involved in excretion. What are they excreting? What are they getting rid of? So the lungs is excreting obviously our carbon dioxide. Our skin, we are going to be losing our ions. We're going to be losing excess water in the form of sweat. And our kidneys, oh, we should have actually done water vapor as well from here. Our kidneys is to do with our excess water. It's to do with our ions that are in excess, and it's to do with our urea. And our liver is the one that is going to produce the urea after deamination has occurred. Carbon dioxide is formed by deamination of excess amino acids. Okay, yep, that's not true because it's not carbon dioxide, it's ammonia. So we want to get rid of that and say it's ammonia that is formed after deamination. Urea is less soluble and less toxic than ammonia. Yep, that's the reason that we actually are going to do the ornithine cycle. So the ornithine cycle is about changing the ammonia into urea, and you do not need to know the actual parts of the ornithine cycle, although if they are going to ask you questions on it, it could be where they've given you information and they want you to then apply the knowledge. And then the last one, breakdown of heme from hemoglobin produces bile pigments that are excreted in the feces. That is correct. It's important to note that the breakdown of the heme from hemoglobin produces the bile. The bile is produced in the hepatocytes, and it is then released into the bile duct. And that's how it gets moved from the liver into the bile duct and down. It gets stored in the gallbladder, so as well, stored in the gallbladder. And what else do I want to say here? Breakdown of heme. The only other thing I would put here is that the iron is going to then be used to make more hemoglobin. So that is able to be used by your bones. So well done. You were absolutely right. You would have got those 2 marks. Okay, I am going to stop there for tonight. Guys, thank you for joining me. I will come back on Wednesday at 9 o'clock and do this a bit more and keep going. Yes, they are correct. They are. And that's why sometimes, yes, you were right to go true, false, true, true. But what we'd actually done by adding a bit more to the revision is we're confirming the knowledge in our head. We are refreshing it. We are doing active recall. And it's really important that you do that more and more. You're very, very welcome, guys. Thank you for joining me. And I look forward to working with you again. Right. Good night, everybody.